All right, guys, we may be live now. Let me put this somewhere convenient. All right, you guys ready? Let's go ahead and start. Okay. All right, guys, let's go ahead and start. So, we've got, I think I've got enough slack here. Yeah. Our exam review. Let's go ahead and start with this. The Proteobacteria, remember? We left off here from the last exam. So I'm just going to flip through and I'm going to highlight some things that may be more important than others to focus on. So as far as the Proteobacterium go, you should probably know what the term oligotropic means, and that alpha Proteobacteria are oligotropic, which means they can survive in low nutrient levels, and they can grow in that. You may want to know about rickettsia, specifically Rickettsia rickettsii is responsible for Rocky Mountain spotted fever. Okay, it's a parasite. What else? Okay. We talked about beta Proteobacteria. You should know about Neisseria, specifically Neisseria gonorrhoeae, which is causative agent of gonorrhea. We talked a little bit about gonorrhea itself. It's called the clap. So just be aware of maybe a little bit about that STD. What else? Gamma Proteobacteria. Oh yes, we talked about the methallotrophs. Okay, you should realize that these ones use methane and other reduced carbon compounds, so they can reduce greenhouse gases, greenhouse emissions, right, by using those. Pseudomonads, we talked about how they're responsible for mineralization. Vibrio-nales, we talked about Vibrio cholerae. You should know that Vibrio cholerae is the causative agent of cholera, and that entails diarrhea. So you don't need to go into huge depth with these, right? Because I know there's a lot of slides and a lot of material. You don't need to get bogged down in all the minutia. It was mainly there for your own information. Yes?
Okay, true, false.
True, false.
Yes, questions.
Yes.
You got me last time.
Got you last time.
Yes, you got me last time.
Yes.
Okay.
My evil ploy worked then.
Yes. How many true, false questions do you think this time around?
I don't recall, and I still have to edit the exam, but it's usually, about 25% of the exam questions or so. Yeah. So then we talked about the Enterobacteriales, and remember this included the family Enterobacteriaceae, which you should be familiar with. It was the family of your unknown. What do we need to know? Obviously, E. coli is the main member here. We talked about the coliform bacteria. Remember that? The non-coliform bacteria and the true pathogenic as well, family members here. Enterobacteria. Let's see, we don't need to know all this minutia here. You see how it's all about capsules, fimbriae, hemolysins, all this stuff. I put this here because it is interesting, but you don't need to spend a lot of time learning about all the virulence factors that we discussed. Okay. Again, the Enterobacteriaceae include coliform bacteria, which are lactose fermenters, non-coliform opportunists, and the true pathogens. Right? We talked about E. coli and how it's a coliform bacteria. It's indicative of fecal contamination in fresh water. If you find E. coli in fresh water, it's a coliform bacteria, and that's a telltale sign that the water source is contaminated. Remember I told you E. coli is a normal microbiota member of your intestines. However, certain strains are pathogenic, right? Like the strain 0157H7, we talked about that. What else do we need to know? I think we talked about Serratia marcescens. One of you had this, one of the groups. You guys had it. It grows with a red pigmentation, which is really interesting. One of you had this one, right? Proteus mirabilis. One of you guys. I know William's team had this. This is known for its swarming behavior. Swarming behavior. Okay. What else? Salmonella, truly pathogenic, right? Salmonella. Okay. Remember that it can lead to disease. Remember that it can be found on eggs and poultry, mainly. Shigella causes dysentery. Shigella dysenteriae is responsible for dysentery due to the Shiga toxin. You may want to remember that. So Shigella dysenteriae, responsible for dysentery. We talked about Yersinia pestis, another family member here, okay, or order member, I should say. Yersinia pestis, which caused the plague. Remember that? And there was bubonic plague, which is not spread person to person. It's spread, from rats, vole, mice, to fleas, fleas to people. So the fleas are the vectors, right? But then if the bacteria were to make it into the lungs, then you could spread it person to person as pneumonic plague, right? So know the difference between bubonic plague and pneumonic plague. Which one can be, speaking of true-false questions, can bubonic plague be spread person to person? No, it's pneumonic plague that is spread person to person. Otherwise, it's rodent, vector, flea, person, right? And this was responsible for the Black Death, right? 70% of Europe died. We talked about, in the class Delta Proteobacteria, we talked about Delovibrio. And what you should know are these are interesting because they behave like a virus. These are bacteria that infect other bacteria, grow through multiple fission, and then lyse the cell to release, right? So their life cycle is somewhat of a bacteria. A virus, a virus, isn't it? So Delovibrio, what should you know about it? Its life cycle is like that of a virus, right? What else do we want to know? We want to know about, in the class Epsilon Proteobacteria, we want to know about Helicobacter, and specifically Helicobacter pylori, which is responsible for ulcers, right? So I'm going to make sure it looks good. Okay. Okay, so, Helicobacter pylori, we want to know for the exam that it's responsible for peptic ulcers. What else? Moving on to these gram-positives now. We talked about the molecules, which are pleomorphic. Remember, these are a class of gram-positives that don't have a cell wall. Okay? And that's why they're pleomorphic, which means they don't have a particular shape. If you don't have a cell wall holding you in a particular orientation, you're not going to have a particular shape. That makes sense, right? What do we want to know about Clostridium? These are important because they're endospore formers. Definitely know for the exam what are the two genuses that include endospore formers. What are they? Bacillus and Clostridium. Definitely know that. That's very important to know because there's only two genus out of all the genus of bacteria out there. Two of them make endospores, right? That's pretty interesting stuff considering there are how many genus of bacteria? Clostridium makes endospores. And we should know that Clostridium botulinum is a causative agent of botulism. Clostridium tetani, tetanus. Okay. Both of those are like, they affect the neurons, right? Clostridium difficile. This one causes, colitis, intestinal issues. This is one you will be struggling with as a nurse, right? Trying to prevent your patients from picking this up at the hospital, right? What else? Bacillus. Remember I said Bacillus, if I could be a bacteria, I'd want to be in the class Bacillus, right? Or Bacilli. Because these guys, not only can they form endospores if the going gets tough, but they can make antibiotics to destroy other competitors too. So that's pretty good. Offense and defense. We talked about Bacillus anthracis. That's the causative agent of anthrax, right? Remember there were three forms of anthrax. Cutaneous, ingested, right? Non-cutaneous, and then inhalation. Inhalation is from weaponized anthrax, right? When they make the spores aerosolized. And they can leave, the cutaneous version can leave these scars. What else? We talked about Staphylococcus, which has these irregular clusters. We talked about Staph epidermidis and Staph aureus. These are, one is, actually both are skin residents. Staph aureus can cause infection. It's a opportunist, right? What else did we talk about? Yeah, staph infections are due to Staphylococcus. A lot of times Staph aureus. You don't need to know all of these virulence factors I've listed here. Again, I put them here for your own information. Or these here, or these here. Okay, or these here. Okay, non-invasive staph infection. This is through usually ingestion. Yes. Oh, yes, thanks for letting me know. That's usually the low battery. Thanks for telling me. Hopefully it's still going. I think it's still, still chugging along. It did? No. It's still counting. Could someone check online and see if it's still, I think it's still going. Yeah, try it again. Hit refresh. Should be. Non-invasive is ingested. Yeah, there you go. Still working. Cutaneous, you get into the skin here. What else? See, it works. Systemic, we talked about toxic shock syndrome. It can cause toxic shock. Then we went on to, we talked about methicillin resistant and vancomycin resistant Staph aureus. What else here? Lactobacillus. We talked about lactobacillus and how it's very important in fermentation. It's also a normal flora on your body. We talked about streptococcus. And how it causes strep throat. Streptococcus pyogenes is responsible for strep throat, so definitely know that. It's also responsible for necrotizing fasciitis, flesh-eating disease, which some of you are going to do, right? 2 of you are going to do, flesh-eating bacteria. That's always a fun one. Make sure you didn't eat dinner right before you come to that. More gram-positives. Here we're talking about the actinomycetes, which are big group of antibiotics. That's what you should know about them. And you should also know that the way they grow is by high fill growth, so filamentous growth. That's why it's hard to distinguish them from mold sometimes because they grow like fuzzy little colonies. That fuzz is the mycelium, which is a network of hyphae. They also disseminate spores, right? Spores get spread out. What else? We talked about Corynebacteriae. including the Corynebacterium, Mycobacterium, Nocardia. C. dip, right? Corynebacter diphtheriae is causative agent of diphtheria. Definitely know that. We talked about Mycobacterium. Oh, very important that you know that the Mycobacterium contain those mycolic acids in their cell wall. This is the group that's considered acid-fast. These are your acid-fast bacteria. Okay, definitely know that. Also, we should know that Mycobacterium tuberculosis is responsible for tuberculosis, and Mycobacterium leprae is responsible for leprosy. And I think a couple of you are doing that. Okay. Mycoplasma pneumoniae obviously cause pneumonia. Is it the only cause of pneumonia? No, no. Many causes for pneumonia, right? That's one of them. Okay, what else? We talked about streptomyces. What do you need to know about streptomyces? You should know it's very common in soil. It's also very prolific source of antibiotics. Okay. Moving on to eukaryotes, the protists. We want to know about these supergroups. With the supergroup excavata, we want to know that the excavata tend to have a feeding groove called the cytosome. An example is Fornicata, and you definitely want to know about Giardia, right? Giardia lamblia, which is really interesting. It causes diarrhea, traveler's diarrhea, right? That's the one to know about. We talked about Euglena, Euglena, which is really interesting. It's photosynthetic. It has a flagella. We talked about trypanosomes, which are whip-like, and they move around with that whip-like tail. You may want to know that Trypanosoma cruzi causes Chagas disease, which, remember, is spread by the kissing beetle, whereas Trypanosoma gambiense causes African sleeping sickness. That one's spread by the tsetse fly. We talked about amoeba. You know, remember that some amoeba are naked, some have a shell. Amoeba proteus is your typical laboratory amoeba that's well studied and everywhere, ubiquitous. We talked about slime molds. I think the only thing to know here is the myxogastria, or the acellular slime molds, whereas the dictyostelia are the cellular slime molds. We talked about chromoaviolata. What do you need to know about these? The apicomplexans have an apical complex, right? And an example is Plasmodium. Dinoflagellates, these guys are responsible for red tides. Ciliates, we talked about Paramecium. These move about with cilia. And the Straminopilla, these are very diverse, aren't they? They include diatoms, algae, oomycetes, all this other stuff. But one thing they have in common is a heterocont flagella, so definitely know that. Diatoms, we talked about these. They've got this shell of silica. And you may want to know, actually, you may want to know that the larger shell is called the epitheca and the smaller shell is called the hypotheca. You may want to know that. We talked about Archaeplastida, which have plastids. These include the algae, green algae, which is photosynthetic, responsible for much of the photosynthesis on Earth. Then we moved on to fungi. Fungi are saprophytes. Definitely know what saprophytes means, right? Feeds on dead organic material, breaks it down. They don't have chlorophyll. What else? We talked about Chytridiomycota, which include the chytrids. These are the simplest fungi, okay? Zygomycota, these include the zygomycetes, and you should know that this family, or the example here that we're interested in is Rhizopus solanifer, which is the common bread mold. We looked at it in the lab. Glomerulomycota, what do you need to know about these? The main thing to know about these is that they are mycorrhizal symbiotes. What does that mean? They grow in a symbiotic, mutualistic symbiotic relationship with plants, plant roots. So that's the important thing to know about them. The Ascomycota, what's the important thing to know here? First of all, that they're called the sac fungi because their spores develop in a sac, right? And that includes these truffles, yeasts, okay, things like that. Basidiomycota, these include the mushrooms. And Microsporidia, these are interesting, and you should know that these are interesting because they're obligate intracellular parasites, right? That makes them different. Okay, and that is it for that chapter. The things I mentioned are the things to really focus on. Any slide that's swimming in detail seems like a lot to memorize, and that is because it is a lot to memorize. So you don't have to memorize all that much detail. You don't need to know it, and because I'm well aware that there are lots of slides, but mainly because I want you to see a lot of things and learn about a lot of things, but not stress you out with the burden of having to memorize all said things, right? Does that make sense? Yeah. Questions about this packet? So from Proteobacteria, which started right after syphilis, right? From Proteobacteria to the end of this packet is on the next exam, and viruses, right? This packet. So let's go through this packet. And again, I'll try to highlight those very important things to know. So obviously know these terms: viruses, viroids, virusoids, prions. What do we need to know about viruses? What's a virion? So virion is the virus particle, complete virus particle. Obviously know that its genome can be diverse. It could be, remember, all different types of stuff: single-strand DNA, double-strand DNA, single-strand RNA, double-strand RNA, and there's even more than that, right? Bacteriophage or phages are viruses that infect bacteria. Archaea can have viruses. Eukarya have a lot of viruses. Okay, what's the capsid? It's the protein coat. Definitely know that. Some viruses have an envelope, some don't, right? And that envelope, they didn't make the envelope, did they? They borrowed the envelope from their host, okay? And then what else? Are they alive? No. No, most scientists will agree that viruses are not alive. Okay, what are protomers? They're protein subunits of the capsid. You should know there are helical capsids. There are icosahedral capsids. I'm not sure if I asked this, but you may want to know that in the icosahedral shape, the pentamers are 5 units, and they're at the vertices, and the hexamers are 6 units, and they're along the edges. And then there are viruses of complex symmetry. Just understand what that means. That means it's not just a simple shape, it's a combination of different shapes, right? Then we talked about envelopes. Remember, envelopes can be very tight, like the rabies virus. Envelopes can be very loose, like the herpes virus. Spikes are proteins on the surface of the virus. A lot of times spikes are there for attaching to your host cells. Remember we talked about in flu, we talked about, remember H1N1, what does the H and N represent? H represents hemagglutinin. Don't forget to add an extra IN at the end of that, hemagglutinin. And what does hemagglutinin do? It says it's involved in viral attachment, okay? And then we also should know neuraminidase. What does that one do? Once the flu is inside the cell, helps it release into the cell, from the vesicle into the cell. Okay, so let's understand what hemagglutinin and neuraminidase do. What else? Remember, the genomes can be segmented. The genomes can be circular, straight. We talked a lot about the viral mechanism here. It starts with attachment, and that includes tropism. Remember that term, tropism? This is the reason why the Berlin patient became immune. Remember that? Because of a bone marrow transplant. In fact, you may want to read up on the Berlin patient and just understand this concept of tropism, how the Berlin patient became immune to the HIV virus strain that he was infected with, and how did the bone marrow transplant help him achieve this immunity? How did he get cured of HIV, right? So be able to explain that. And there's lots of resources online you could look up. Your book also, I believe, mentions his story, and you could read about it, right? You know, that might be something to understand. Okay, remember, if a virus cannot attach to your cells, you're immune to that virus, right? Some viruses infect all kinds of different cells and all kinds of different animals. Some viruses infect a specific tissue on a specific species of animal. You know, it's all to do with the tropism of the virus. And you may want to know, again, about the Berlin patient. What is CCR5, delta 32? What does that mean? How did the whole process work? That might be something interesting to understand. And then we talked about entry. This is pretty self-explanatory. We talked about synthesis. This is pretty straightforward. Assembly, straightforward, and released. Remember, non-enveloped viruses usually lyse the cell, whereas enveloped viruses usually bud, right? And don't lyse the cell. But you know, there are exceptions. We talked about virulent phage versus temperate phage. Virulent phage have one life cycle option. It's get in, destroy, get out, right? Temperate phage have two options. They can either be virulent or lytic, or they can be lysogenic, right? Lysogenic means incorporate the DNA into the host, live there for a while until the conditions worsen, then switch to lytic to escape, right? To produce more virus and escape. Okay, we talked about eukaryotes as well. We talked about eukaryotic cells. Oh, that happened again. Okay, hopefully that didn't cause too much of a hiccup. We talked about eukaryotic cells as well. Just remember, in eukaryotic cells, you can have multiple outcomes as well. It can either destroy the cell, it can be a chronic infection, it can cause cancer, right? Viruses can cause cancer. Anything that disrupts your DNA, causes mutation, changes your DNA, could potentially give you cancer, right? We talked about that. Okay. These were the two mechanisms by which viruses can give you cancer. Either they can bring an oncogene into the cell, or they can change one of your genes. Yeah?
To back up on that infection of eukaryotic cells, do we need to know like tumor, neoplasia, anaphasia?
Not necessarily, no. Just know that it can lead to cancer.
Okay.
You don't need to know this list. Just be aware that there are viruses and certain strains of those viruses which can lead to cancer by disrupting what? By disrupting your proto-oncogenes or by introducing oncogenes, right? What else? We talked about viroids, and what you should know about these is that they do not encode gene products, so there's no genes on there that make proteins. That's good to know. They infect plants. And how do they work? With silencing, RNA silencing. Okay, those are the important things to understand about viroids. Virusoids, on the other hand, infect animals. Like they could, you know, for example, hepatitis D infects people, but it's just RNA, and it does code. Look, it does code for gene products. It does make protein, right? And remember, it piggybacks on hepatitis B virus. Prions, remember, can lead to these series of diseases. I think one group's doing Kuru, one group's doing Creutzfeldt-Jakob disease, okay, CJD. It's interesting to know, and it's just made of protein, and the mechanism is protein unfolding, right? The prion protein is misfolded. It can cause a cascade of misfolding in your body, which can lead to massive brain damage. And then we talked about viral classification, specifically the David Baltimore system, or the Baltimore system. You should know about group 1, 2, 3, 4, 5, 6, 7. Okay? With each of these groups, you should know how the virus makes more of its genome and how it makes more of its proteins, right? Double-strand DNA viruses use your enzymes, right, to make more of its genome. And it uses your ribosome to make more of its proteins, right? Very straightforward stuff because it's double-strand DNA. You don't need to sit here and memorize that herpes virus and pox virus are examples. That's a little excessive, right? Double-strand or single-strand DNA, you should know that that's group 2, and there's just one extra step. It's rolling, it converts the single-strand DNA to double-strand first, and then it makes more of its genome. It also makes proteins. Group 3, what do you need to know about this? Oh, the important thing to know about group 3 is not only this double-strand RNA genome, but also that the positive strand can serve as mRNA, right? So this virus can either bring with it, remember the RNA-dependent RNA polymerase, right? Or it could just code for it in the cell, right? Like the ribosome can just make it for it, right? What about this one? The group 4, you should know that same thing, right? These have single-strand, positive-strand RNA, which can serve as mRNA. So again, these viruses can bring with them that enzyme, RNA-dependent RNA polymerase, or just code for it once there, and your ribosomes will make it for it, right? So it can make more of its genome, right? But what about group 5? Group 5 is not the same. It's negative strand RNA, which isn't so easy, is it? Here, it must bring with it that enzyme. It cannot code for it once there. That's the thing to know. Retroviruses, what do you need to know here? What you need to know here is that these viruses, they have to use not just a host of other enzymes, but they have to bring this special enzyme, reverse transcriptase, with them. Okay, remember, these are the ones that need reverse transcriptase because they revert their RNA to DNA. Okay. And then group 7. Group 7, you need to know that these have a strange double-stranded circular DNA. One has a gap, one has a nick, and this involves formation of a pregenome, okay, in order to make another copy of itself. Okay, you don't need to know it in more detail than that. Let me see here.